Let us take question number 8 now, which says, what are the characteristics of prokaryotic cells? This is our question 8. Characteristics of prokaryotic cells. Prokaryotic cells, that means a typical bacterial cell, plasma membrane, cell wall outside it, there is no organelle. So, before we write the points, we should draw. In biology, drawing the diagrams, labeling them properly help a great extent. Reason, just when the examiner sees this diagram properly labeled, the person gets an idea that the student knows everything. So, it is always good to supplement your answer with a diagram. Though in the question it is not mentioned draw. So, but quickly we can draw a simple diagram. Plasma membrane with its infolds, which are known as mesosomes. Then there is a cell wall outside it, which is made up of peptidoglycan. And the same points we will write down in the characteristics. The genetic material is in the form of a circular DNA, cytoplasm or protoplasm, in which there are ribosomes, which are 70S type. So, now just quickly label all these things. This is plasma membrane. These structures which are here, folds, are mesosomes. Ribosomes, the 70S type. This is DNA, and this is the cell wall. We can also draw a flagellum. So, now when it comes to writing the characteristics, what all characteristics we would write? These prokaryotic cells, they exist in multiple shapes. So, variable shapes. They do not have membrane bound structures or organelle. The nucleus is absent. Only circular genetic material is present, which is known as nucleoid. So, DNA as nucleoid. They have ribosomes, which are 70S type. Then outside the plasma membrane there is a cell wall. So, they have cell wall. And what is the cell wall made up of? It is made up of a substance called peptidoglycan. They may have flagella, they may not have flagella. If they have flagella, then the number of flagella is variable. It can be one flagellum or many. So, this is variable. No membrane bound structure. So, all these things would become the characteristics of a prokaryotic cell. So, our question was, what are the characteristics of prokaryotic cells? So, we have drawn it, plus we have written down all the points which are there in a prokaryotic cell. Question number 9. Ninth question is, multicellular organisms have division of labor. Explain. They have given us a statement and they want us to explain that. The statement is that multicellular organisms show division of labor. What is first, let us understand what is meant by division of labor. Division of labor basically means when the different jobs are assigned to different structures or organs or systems. Like in case of our body, the job of respiration is done by respiratory system, transporting gases and nutrients is done by the circulatory system, removal of waste is done by excretory system. Though all these systems, they work in coordination with each other. But why is this required? This kind of division of labor is not needed in a unicellular organism because one cell is capable of doing all these things with the help of organelle. As the number of cells becomes bigger and bigger, that means when they become billions and trillions, all the cells are not in contact with the surface. So every cell requires oxygen. If it is a multicellular organism, are all the cells in contact with oxygen or air? Only outer cells are. If diffusion has to take place, if the innermost cell, say for example, this is a structure where there are multiple cells and only outer cells are in contact with oxygen or air. How will this cell get oxygen for aerobic breakdown of food? Only simple process, that diffusion from outside to this cell, from this cell to the next, next to next and so on. So by the time it reaches the innermost cell, the oxygen content or the nutrient content is going to be negligible. Plus it's going to take a long time because diffusion is a passive slow process. So if the work is divided, that one system does one thing or one group of cells do one thing and other group does other thing, and these two groups, they don't worry about the other's job. Only thing is there is coordination. That means the digestive system need not worry about respiration. It knows it will get its share of oxygen. Similarly, respiratory system need not worry about how will I get nourishment. So they don't worry about others' job, but there is a coordination between all the systems. So division of labor is required in multicellular organisms to take care of all the functions, and there are different groups of organs which are assigned these functions. So this is for proper maintenance of the life process. Question number 10. Cell is the basic unit of life. Discuss in brief. Cell, basic unit of life. This is actually the title of our chapter also. What is meant by unit of life? Unit of life is the smallest thing which is performing all those functions which an organism can. So what are those things which an organism shows? Growth. All organisms show growth. Respiration, digestion, excretion, reproduction. These are all life processes, and a single cell can show all these processes. Cells grow, cells need oxygen, they respire, they digest the food, they excrete the waste material, they reproduce also. So it is the smallest unit which shows all the processes of life, and that is why it is known as the basic unit of life. Question number 11. 11th question is, what are nuclear pores? State their function. When we were studying the structure of nucleus, we said that the nuclear membrane is not continuous. There are some perforations. These perforations are known as the nuclear pores. We also made the detailed structure. The nuclear membrane is a double membrane structure. So there is an outer membrane and inner membrane. This is the outer part, this is inner, and between these two membranes, at the junction of these two parts, there is an opening. This is known as the nuclear pore. But this nuclear pore is not a gap or an opening like this. There is a complex which is fitted here, and this complex is known as the pore complex. In the center, there is a protein which acts as a channel protein. So basically, pore is for entry or exit of certain things, but it is not just through and through structure from where anything can go in and out. Now, what is its function? It is meant for certain things to go out and few things to come in. What are the things which would go out inside the nucleus? The DNA makes RNA, mRNA, when transcription takes place. So this mRNA goes out or moves out through this pore. RNA, other RNAs like ribosomal RNAs, they also go out of this. The substances which are needed by the cells, like the raw material, all those things would come in through this pore. So it is basically for exchange or transport of substances which are required for the nucleus. Plus the main things which will go in and out are the proteins and RNAs, which are either synthesized inside the nucleus and they have to go out. So this is nuclear pore, and its function is for exchange or transport of substances. 12th question. Both lysosomes and vacuoles are endomembrane structures, yet they differ in terms of their functions. When we made the structure of a lysosome, we made a circular, simple, membrane bound structure. When we made lysosome and when we made a vacuole, at that time also we made a circular membrane bound structure. Both these membranes are typical plasma membrane like structures. In case of vacuole, the membrane is known as tonoplast. Otherwise it is typical membrane. Now what is inside makes them different. Inside the lysosomes are present hydrolytic enzymes like proteases, lipases, amylases, protein digesting proteases, lipases, lipid digesting, and amylases. All three are hydrolytic enzymes. They help in breakdown of food. What is present in the vacuole? There are multiple things. We talked about food vacuole, so they can store food. So it would be a food vacuole if some kind of food is present. It could be air vacuole. It could be excretory vacuole when some waste is accumulated. And it could be contractile vacuoles, which is helping in osmoregulation. So basically their functions are different, though both have a simple membrane and they are pinched off from Golgi body. So structurally they are same, but functionally they are different because they contain different things inside. Lysosomes have enzymes, and we talked of suicidal bags also. So they help in digestion of food as well as damaged organelle or the cell itself. Whereas vacuoles are normally storage structures. Either they store food, air, waste, or they can help in regulation of the osmotic pressure of the cells, that is osmoregulation. So functionally they are different because their contents inside them are different. The 13th question is, describe the structure of following with the help of labeled diagrams. So, let us draw the diagrams of these two structures, that is nucleus and centrosome. Question number 13, describe the structure of the following with the help of a labeled diagram. So, first we will have to draw and then we will have to explain the structure. So, this is our question number 13. The two structures which we are required to describe and draw, one is nucleus, another is centrosome. So, let us take nucleus first. Nucleus is a double membrane structure, normally spherical, but there are other shapes also which exist. The outer nuclear membrane continues with endoplasmic reticulum. So, at some places we can also draw this extension into endoplasmic reticulum. Double membrane structure, outer membrane, inner membrane. Between the two membranes there is a space which is known as the perinuclear space or intermembrane space. The nuclear membrane has pores. So, these pores are known as nucleopores or nuclear pore. The inner area is filled with a jelly-like material which is known as nucleoplasm, which is also known as karyoplasm. Karyon is the term given to nucleus, so karyo term is also used. For example, these membranes can also be termed as karyotheca, the inner and outer. Nucleoplasm can be termed as karyoplasm. And inside this nucleoplasm are the thread-like structures which are chromatin fibers. Along with this, every cell has at least one such dark stained structure which is called nucleolus. So, simple structure with all labels. Now, we have to describe this structure. So, what all points would be there? That nucleus is a double membrane structure, the outer membrane extends into ER, the inner material is known as nucleoplasm which is rich in enzymes, divalent ions. Nucleoplasm is rich in enzymes which are required for various processes, like DNA is going to replicate, then from that DNA mRNA will be formed. So, replication of DNA, transcription, so enzymes for DNA replication and transcription. So, all these enzymes are present in nucleoplasm, plus it has divalent ions. These ions, especially magnesium and manganese ions, very important, they are as activators of most of these enzymes. Plus, the this nucleolus is the site for ribosome synthesis. So, this nucleolus is important. Chromatin, which is basically chromosome but thinned out structures. So, in this case only fiber like, we do not see the chromosome structure, and all the activities of the cell are controlled by nucleus. What are the activities? Everything, that is all processes are regulated by this, plus it helps in inheritance of characteristics. So, the genetic material which is present in the nucleus is transferred to the offspring. So, it also helps in inheritance of genetic material. Then the second structure they have asked is centrosome. So this was nucleus, then centrosome. When we talk of centrosome, it is a structure which is found only in animal cells, or the structure which we talked of, we said there are two centrioles surrounded by a clear cytoplasmic area and peritubular fibers. This whole structure is known as centrosome. Now, in centrosome, the most important part is the centriole. There are two centrioles, or the centriole rod-like structures, which are placed at right angles to each other. So, if one is vertical, the other is going to be horizontal, and we have seen the structure of the centriole also. There are 9 peritubular fibers, the tubulin ones. In the center, there is a hub. These peritubular fibers, they are in the form of the triple units or triplets. Then from everywhere there is a radial spoke arising from the hub. There is a thickening at the base of it. All these structures are made up of tubulin proteins. Then A, B and C, this is how we labeled it. C of everyone is connected with A of other with the help of AC linkers. These are AC linkers. So the arrangement of microtubules in centrioles is 9 plus 0. There is nothing in the middle. The tubules are only in the peripheral part, and there are 9 in the peripheral part. So just go through that detailed structure, and we have to draw the structure. So here basically the structures are asked, and then on the basis of those things we have to describe them. 14th question, the last question on this is, what is centromere? How does the position of centromere form the basis of classification of centrosome? Support your answer with a diagram showing the position of centromere on different types of chromosomes. Centromeres are those primary constrictions. This is question number 14. So when we make the structure of a chromosome, we make these chromatids, and these chromatids, they show a constriction in the middle. This is known as the primary constriction or centromere. On the outer side of this centromere, there is a protein sheath which is known as kinetochore. And to this kinetochore are attached the spindle fibers during cell division. So how is the position of this structure? Gives us different types of chromosomes. We talked of 4. Metacentric. Metacentric are ones in which the lengths of these two chromatids are equal. That means the centromere is centrally placed. If, or let me just draw all 4 here. This is metacentric. In this metacentric one, both the arms are of same length. The second one is one arm longer as compared to other. So one chromatid, one arm is going to be long, other is going to be short. So this is metacentric. This one is submetacentric. If this arm, shorter arm, is very, very short. So if this is the centromere, this is arm, and the other arm is very long, then this type of chromosome is known as acrocentric. And the 4th one is when the centromere is terminal. That means only one arm is there. Centromere is at the tip of that arm and the other arm is totally missing, then these type of chromosomes are known as telocentric. When we were discussing this, we also discussed that these chromosomes are represented by some alphabets. Metacentric is drawn as this, that is alphabet K. Actually this line is not drawn, but this is for K. Submetacentric as L, acrocentric as J, and telocentric as I. So these are representations of these chromosomes. The difference in these four is due to the position of the centromere. If it is central, two arms of the same length, metacentric, one arm long, other arm short, then submetacentric. The short arm is very, very short and the long arm is very long, then acrocentric, and if the centromere is terminal, then it is telocentric. So there were 14 questions after this chapter and we have discussed all 14. So while writing the answer, 2-3 things which we have to keep in mind. Number one, if a question has multiple parts, we need to answer all the parts individually by giving it maybe in different paragraphs or different lines so that we are able to score for each and every part. Always support your answer with a diagram, because that helps the examiner understand things better. Make the diagram clear, label it properly, and go through the notes very, very carefully. All important points are in the notes in our lectures. Understand them, revise them, and I am sure you will be all set with this particular chapter.